All right, go ahead, Krishna.
Hello, everybody. Welcome to the seminar this afternoon. Before I briefly introduce the speaker, I am Dr. Krishna Kaluri, alternate counselor at the Southern California section of the American Chemical Society. And this particular seminar is funded by the Local Section Activities Committee, LSAC, Innovative Projects Grant. And our local section is very thankful to LSAC for generously funding us to organize seminars like this. And I'll give a very brief introduction of today's speaker, Phyllis Frankel. I won't touch upon what is in the flyer already, but I will very briefly mention that she is a research scientist at the Massachusetts Institute of Technology in the Department of Chemical Engineering with support from Mechanical Engineering. Phyllis's work have been profiled in the New York Times, the magazine called Wired, Life, Boston Globe, Washington Post, the Chronicle of Higher Education, National Public Radio's All Things Considered, and Science Friday, and various European publications. A limited edition of photographs are included in a number of corporate and private collections, and her work has been exhibited throughout the United States and Europe, including at the Kennedy Center Arts Summit, the Museum of Modern Art's 2008 exhibition, New York Hall of Science, and along the Champs-Élysées in Paris, and as a traveling exhibition supported by Bracco Imaging Group in Italy, including Genova, Rome, Naples, Perugia, Milano, and the New York University's Casa Italiana Zerilli Marimo. Her work was featured in the 2016 MIT Museum exhibition, Images of Discovery, Communicating Science Through Photography. And before I hand over the mic to Phyllis, I just wanted to quote one sentence from her. I believe that we who are privileged to see science's splendor, who image it, diagram it, model it, graph it, and compose its data, can turn the world around, dazzling it with what inspires and nourishes our thinking. And she added, our goal must be to share the visual richness of our world to make it accessible. Now over to Phyllis for her presentation.
Hi, thanks so much, Krishna. It's a real joy to be with you all. I was really delighted to get the invitation, and I want to thank you. I was mentioning before, I'm sort of on a mission, sort of, so to speak, in that I'm going to try to make a case for you all that making pictures, um, oh, there we go, which are basically making representations, is making more than pretty pictures. There's something deeper involved with the process, not only for the person making the representations, but for the people viewing it. That there's something deep going on that, in my opinion, it should be part of your training if you're a research scientist or an engineer. That's the case I'm going to try to make in this presentation, and we'll have questions as we mentioned. Daniel mentioned in the chat after, and I'm eager to talk with you afterwards if something comes to mind. Just don't put it in the chat because I get confused, but afterwards we could do that. So keep in mind that this is going to sound silly, but representations are representations. They are not the thing. So there is some kind of thinking, some kind of translation that goes on when you're making a representation. The process itself, frankly, and I'm not embarrassed to say, brings attention to your work. I love the fact that I'm involved with very wonderful, groundbreaking work, and I'm privileged to be part of that community. And I do help to bring attention. Years ago, I made this image of nanocrystals, quantum dots, from Moonji Buendi's lab. And sure enough, he got the Nobel Prize last year. Yeah, yes, of course, I had nothing to do with his getting a Nobel Prize. But the fact is that this image kind of was shown all over the place, and I was helping to bring attention. What you're seeing is um, nanocrystals, cadmium selenide, in vials, but the size of the nanocrystals change. So they fluoresce at different wavelengths under UV light. That's what you're seeing. And the fact is it is a document of the work that he was doing. He didn't like that picture. He wanted, because I took it in such a way, you might consider how I did it. I laid the vials down. Let me see if I could go back. Yeah. The vials were lying down on a table and the camera is above. So I thought by having it this way, it shows the air bubbles, but he thought it was in the way. And, you know, I could argue either way. But the bottom line is, so I reshot it, and that in fact got on the cover of JPhysChem, which never has images on the cover, but this image is in fact a document of data. And it is in fact more than just a pretty picture. And it helps bring attention. Believe me, these researchers, Bob Langer does not need me to bring attention to his work, but I took a very simple image of the work and it got all over the place. And it was really exciting because I was helping to bring attention. I also like to think that the images that I make communicates the science in a way that text doesn't come close to communicating. This is an image that came out of the laboratory from many years ago when I first started this whole new adventure. This is out of George Whiteside's lab, and you're looking at something called a self-assembled monolayer. And what's going on here is that these are hydrophobic lines that are creating a pattern. And so when you drop water on this surface, the water spreads and stops at the hydrophobic lines. That is the purpose of this work. It's showing we have control over something called a self-assembled monolayer. This is, by the way, this is water dropped, and this measures about 4 millimeters across. So this is the way it appeared in the article. And this is the image that I made. What I did was I asked them for a different sample, for example, a more interesting grid. I asked them if I add some colored dye into the water before I dropped it on the sample, would that be okay? Because the idea is that we want to show that this hydrophobic line is preventing the water from blending together. They are not showing that. I believe that I'm showing that better. This also measures 4 millimeters. And so comparing that, I'd like to think you agree that it's more communicative. And yes, I'm showing off a little bit because it got the cover of Science, which I was delighted about. But in the end, it is about communication. And it was really one of my first assignments that changed my life around. I came back to science after being away from it for many, many years. Now here, Alice Nasto created this material that she wanted to study insulation, and she fabricated material that was similar to sea otter. And this is the stuff that she studied. She made the image as well. I took it from her. She was in mechanical engineering, and I took this identical thing and I folded it and made a very different image, which I hope you agree is something that you'd want to look at. At least I hope you feel that way. It was just about making an image that was more than good enough. It was very simple. I just folded it. I lit it a certain way. And I think it works. I also feel that making the right image or graphic clarifies the science, not only for somebody viewing it, but for the person making the representation. So in this situation, this is a submission to NSF, and there was a space, they specifically asked for a space, a graphic space to explain what the executive summary was about. Well, I looked at that, and to me, it was a little overwhelming. And so I took a number of other pieces, made it simpler. And when you come to this page as a reviewer, maybe you look at this one more because it's simpler. There's white space. You're not jamming in every conceivable idea. You're simple. This is the ongoing lesson that I'm trying to teach students. Simplify, simplify, and edit. It's got to happen. What we do on campus is we create workshops. And so the idea of creating graphics and figures it's collaborative. We have a wonderful workshop running that they bring in their graphics, or their part of their submission to journals. And as a group, we discuss it, and we always find other ways to refine the image or the graphics. And it really is very helpful as we look at each other's work. The issue is with all of researchers, it's something that I find very exciting, is that the challenges are universal. What kind of color to use, how to organize the pieces. It's all the same issue. And having workshops with different disciplines together is really the best workshop. But for example, here's an image that I deleted the labels, and I'm making a point here when I introduce it to the workshop participants. I ask them, what do they think they're looking at? And I'll let you look at it for a moment, and if we were able to talk to each other, I would get your feedback. But basically, I will bet you that this is reminding you of something that you are working on, or in the field that you're working on, or the scale that you're working on. You might see this as some biological information. But if you were in this world, you would recognize that this is the subsurface structure below a sunspot. This is obviously a visualization of data. But what I'm getting at is we all sort of bring in our own baggage, so to speak. The images become Rorschach tests in a way, but yet at the same time, it's a means of talking to each other, which is what we're trying to do. We put graphics students together with science students. We had a program called Picturing to Learn, where the high school students were, the high school teachers were talking to each other about how to visualize concepts that they were teaching. We brought it to Harlem, and these science teachers were explaining to each other in the graphic. It's a means of speaking with each other, and even, may I say, correcting each other, because through a picture, people are more comfortable in speaking with each other. Making the right representation, I'm sorry to be so crude, but I'm going to do it. It really helps with funding. When you come up with an honest depiction of your work and it is aesthetically pleasing, people will look at it. I am showing off again. I apologize, but I am trying to make a point here. These are all covers that I've gotten over the years, and it helps. It helps with funding. If it doesn't give you all the money you want, but it brings attention and therefore helps with funding. And the biggest soapbox that I'm on is that making the right image or graphic makes science accessible to the public. Yes, I got a little silly here, but I feel very deeply that we in the research community have got to speak to the public. and tell them what we're doing and engage them in our conversations. And I believe that making the right kind of image or graphic can be a powerful tool to open the window to our research. So we'll just briefly run through photography, which is where what I really started doing, and now I'm getting just as much into graphics. But for example, if any of you are interested in any of this, I encourage you to go to my course online. If you go to OpenCourseWare, that's OCW, and just put in my name, you could see all the tutorials. They're very good. I mean, I'm told you don't see me at all. It's all voiceover and lots of images and how I start from here to there. Um, uh, if you go to edX, you have to sign up for edX, and you might not want to do that. But from that, I have a number of books out, but the latest books are called The Visual Elements. The first element is in photography. It's a handbook, and I think it's quite helpful, and it's small. It literally is a book that you can carry around. And so, for example, in the book, I talk about using a flatbed scanner. You're not going to believe what you could do with a good flatbed scanner. For example, I put on the scanner some 3D printed material of John Hart's work. I wanted to show the flexibility of it, so I folded it, and this is a scanned image. This is an image I made a while ago of something called a euplectella. It's about 8 inches long from Joanna Eisenberg's lab. Now watch as I zoom into the image. I'm not making a new image. I'm zooming into a very high-res image. And do you see these fibers? These fibers are 50 microns to show you the resolution that you can get on a scanner. It's kind of mind-blowing. You could put your tomatoes on a scanner. And when I ran the course, one of the students decided to create a application where it tells the scanner to take an image every 15 seconds. And he put some supersaturated sodium chloride on the scanner, and look and behold, you have a movie of it nucleating out. It was really quite wonderful. And you could think of things that you could do with a scanner. It's amazing. These are devices that have micro needles that perhaps eventually will uh, attach some vaccine to the skin. That was the purpose. This is another image on the scanner of John Rogers' work, and notice it's buckling a little bit. All I did was put a book on over the scanner's cover, and it flattened out. I mean, this image took all of 10 seconds. It's nuts that you could get a very high resolution image. Here, I put a device from Mehmet's lab, and you see, you're not seeing the detail that I know is there. If you look very carefully, I don't know if you could see it on your screens, but there's subtle material going on in the background. So I took the entire image and fixed it in the histogram. And this is the image that I wound up with. It's just basically manipulating the image, but I am permitted to do that because I did it to the entire image. You're not permitted to take a piece of the image and increase the contrast. If you're going to do anything like this, this is permitted by the journals. But it's kind of wonderful how much you could see that the scanner does in fact capture. And of course, I talk about using a camera. I talk about if you have to image a device, try to image two devices instead. First of all, it shows the ability that you can produce two devices, but also compositionally, it is more interesting. You can make another image on the scanner and you can invert it in Photoshop, and you just get a whole different picture. And both of them, I think, are just as good. I talk about using a phone, which I have become used to now. At the very beginning, I wouldn't frankly take it seriously, but now I do. I did get the cover of Nature with a phone image. Now, in this situation, um, I, I, I formatted it. I took the image without needing to enlarge it. This is the entire image that I took on my phone because I knew that it had to be a high resolution image, but it still is not there yet. I'll show that to you in a minute. This is a scanned image of Sangeeta's work. She's growing E. coli in a petri dish under 4 different situations, environments. That's the image I took on the scanner. This is the image I took with my phone, and I'm overlapping them so you see that the file size is still not right. It is getting better, and I bet it's even better than we're seeing in the shops. I bet it's just as high as, not as high as a scanner. No, it's never going to be that high. But just showing you that the file size, the information is not quite there. So if you take a detail of those previous images, this is the scanner image of a detail of the colonies, and this is the phone image. You're actually seeing the pixels. It's not there, but it's getting there. And so it's very exciting. What I also like to do in photography is putting it together. I'm not an artist. I never pretend to be, but I do know how to take pictures. So I can't draw, but what I do is take pieces of images and put them together to try to create a metaphor or a photo illustration. So, for example, for this assignment, um, I took not a very good picture, but I wanted to show it to the art director at Nature. I sort of liked the way it could be imaged in a cover. And I went back and so what I did was I overlaid it with an illustration of graphene, which is part of the story. And then I sent it to Kelly Krause, who went back and forth with me on what we could do. I made more images like what she asked for. And in the end, they brought it up a notch or two and made a very interesting metaphor for something called remote epitaxy in the presence of graphene. In other words, all of these pieces are photographs that an art person, an illustrator, Nick, absolutely did things that I could not do. Here's something that I did do. I took the background of an image that I made a while ago because the researcher liked it. I then bought the rights of an SEM of E. coli. I made a drop that came from another image. I made a drop. It happened to be of water. I took the outside of that drop and I warped it. That's the outside of that drop. And I colored it and I inserted the E. coli into the warped drop, getting this, and eventually put them all together. That got on the cover of JACS. What she's doing here, Dr. First, is she's protecting the E. coli. That's the bottom line. That's the punchline of this image. And so I put it all together, and I was delighted that they went for it. And it's something I enjoy doing because I can't draw. What can I say? Here's that background that you just saw. This is an image I made for a book that George Whitesides and I published called On the Surface of Things. We wanted to talk about what a drop of water is. And so this is a syringe that has a drop of water. And if you look carefully, do you see that the background is actually coming into focus? The background is, let's see, yeah, the background is this. It's a palette of watercolor. And of course, it's out of focus because I'm using a 105 lens, which is what I knew was going to happen. And so that background blended together. And so I took the drop. I started playing around with extending it a little bit. I put a syringe on the flatbed scanner. I inserted the syringe in the drop. I then took their images of fluorescing pancreatic cells, inserted it in the drop as well, and did a little shadows and stuff. And eventually that got on the cover of Nature Medicine. What they're doing is they're alginating the pancreatic cells to, once again, in this case, protecting the cells if they were ever going to be infused into an animal so that the macrophages would not eat them up. That's the idea. But it's a metaphor, of course. It's not the thing. It's creating a representation of a concept. And so the workshops that we run are not in photography, but they really are about design and designing figures and even posters that I'm not going to get into today. But the handbook that is also out, again, a part of the series is the visual elements, and design is in fact one of the elements. And what I have come to see throughout all my one-on-one conversations with the researchers and the workshops is you know too much when you get your figure together. You want to show everything. You want to show every conceivable data point and what you did. And you can't. It's too much. You've got to figure out what you could delete by asking yourself this question: what is the first thing you want us to see? We can't see it all. What is the primary? What is your hierarchy of information? What is the primary thing that I have to get from the figure? So we could do this with different tools. We could use color, which is a very powerful tool that is overused, in my opinion. When I see these figures, researchers just put every conceivable color they want, they like, and they think it helps, and it doesn't. So in this is an interesting case from a while ago out of Klaus Jensen's lab. This is an illustration of a device that has, these are gels. These colored things are gels. And when they address a current through the device, the gels change pH. Now, when I first looked at this, I said, wait a minute. If you're talking about pH, why is this green? And they said, well, it shouldn't be green. So here we just changed it so that the gel that they want to address with that particular area is in fact the right pH in terms of how we read acid and base. And so it's a small change, but the fact is color has to be used properly. Here, Antonio wanted us to pay attention to the deposition on the liposomes. That's what we're supposed to be looking at and how it happens. If in fact we should pay attention to what is being deposited, why are we coloring the unnecessary liposome? This is something that I go on and on with. Don't be afraid of grayscale. This way we get to see it, or, we're not distracted, especially by using the same color. This was previously red, and this is red. When you see two colors together, you make a connection, and we're not going to want to do that with this. The same thing here, the grafting modification with polyurethane, and then they added this graft. But again, if you want us to see the functionalized areas, get rid of the backbone, which is not necessary in this particular image. Here, this is out of Pecco's class on sports mechanics. There are the data points, and of course, this is about showing it on the field, the data points. And it's a natural thing to show the field in green because it is, but do we have to use green? Do we see the data points better if they're gray? And you might argue with me back and forth. That's fine. I just want to introduce the possibility of getting rid of certain colors when they're not necessary. This is an example out of a lab where these are two visualizations of explosions, two explosions. And the researcher wanted us to only pay attention to that area that is outlined, to compare this area to this area. And so we say, you want us to see that? Well, get rid of the other colors. It's not necessary. It's in the way. And this way we can see what you want us to see. And this is from many years ago that it's called a quantum corral. Don Eigler and his colleagues literally deposited atoms on a substrate. Amazing. They colored it this way. These, of course, it's numerical, which they translated into a visual. And when I talked to researchers about this, was quite a feat, but what I did not know was that the quantum effects going on within the corral are just as important as the atoms themselves. And so I was suggesting to him, and he didn't go for it, but it's fine, to get rid of the color altogether. Because if you want to equalize the information to say, we're doing this and we're doing this, you don't need color at all. And maybe at some point somebody wants to argue with me about this, and that'll be fine. By the way, I have to say this is very important. Anything that I'm saying to you is my opinion. I have no data on this. I have not done a study whether I'm right about this. It's not what I do, and maybe somebody else will do it. But remember, everything I'm saying is opinion. And here, Tonio wanted us, interestingly, he wanted us to pay attention to the buffer layer. But look how he's coloring everything else but the buffer layer. So all I did was say, look, you want us to look at the buffer layer? Just color the buffer layer, and in the TEM, make it the same color so that we know that you're talking about the same thing. That's very important. When you see red somewhere and you see it elsewhere, we tend to make the connection. And sometimes you make a mistake and you don't want to make that connection. And I hope you agree it's better. And and this basically it's a use of one color with different saturations. This is COVID over the year. And it's so much it's easy to read that the darker this color happens, we see the months where the numbers were very high. Color can be incredibly powerful. We use labeling is another, and one thing I want to encourage you is to please stop using slanted arrows. It messes up the image. Try to straighten out your arrows, either make them horizontal or vertical, or maybe do something like this if the layout doesn't work. It just is a neater way of labeling. Why some people label their material, their stuff with a framed white label, I don't understand. I see this a lot. Don't do it. You're adding yet another thing to pay attention to. Just do a nice, simple labeling, smack on the image. And I also want to encourage you to use all lowercase letters. And you might argue with that as well, and that's fine. But it's just a cleaner way of showing. Basically, you're just labeling the pieces. You want us to be it should be simple and elegant. We talk about the tool of composition. This Ivan pathways are a killer to show. He composed his pathways like this, and he included all kinds of icons, colored icons of cells. He also had some colored titles. We work together on this, and I basically turned the whole thing around. It's still pretty busy, but a little less busy. Just simplifying the entire thing, editing out all those big colored cell types that I'm not sure we need if it really helps to see shapes. but he wanted it. But in any event, I hope you agree it's better. Here is the issue of what I call real estate. You have various pieces of the figure, and they're all pretty much the same size. In fact, maybe this is a little bigger. If you want us to pay attention to the differential expression, which is the most important part of the research, then help us look. What is the first thing you want us to see? And it's this. So make that larger and consider making the others smaller. The organization as well. This is so crammed with it, with icons and words, and I think that this is a better rendition of it. I spread it out more. I got rid of the red sentences. It's important for the most part not to label text with red. Just stay with black. The rest of it can be colored. And as I see this again, I still think it's better. But you might disagree, and that's fine, I promise. So, and this is also from quite a while ago from George Whiteside's lab. This was a submission to show the ability to fabricate these self-assembled pieces. And so they put information on it all, and you sort of don't know where to look. All I did was I took their original images. I didn't take any of these pictures, and I laid it out according to size so that there was some sort of ability to make some sort of story out of this. And I would have later included letters in these that would connect with the pieces, and the caption would then have A, B, C, and D, etc. It's just a question of ordering them in some sort of scheme so that we understand what they're doing. Layering is another tool that it could be very powerful. Jay Pasakoff, who's no longer with us, this was his image. I think it's very good. Angela DePace and I wrote a book called Visual Strategies, and we thought that this was a little better. That maybe, yes, he's showing us with the arrows that he's taking this from here. What we're doing here is outlining it exactly the way the image finally wound up. You know, this is a mosaic of images. That can be argued as well, but I thought you'd be interested in how layering can be helpful. Sometimes you can overlay if your axes are the same, you can overlay them, and that makes for a much easier way of reading it. The same with this one, the axes are the same, and so overlaying them, you don't always have that luck, but that it definitely is something you should consider. I'm including this because it's a point that I'm trying to make for you all. If you were in Boston and taking what we call the T to outbound to Alewife is the stop, and if you're at Kendall, which is where MIT is, and so if I were going outbound, I only am interested in what the outbound stops were. So the designer of this poster did not include the previous stops. They edited it brilliantly. And this is what I'm trying to explain to you. By the way, take a look at other things when you walk around your own neighborhood and wherever you are. Take a look at the way people are designing directions. And maybe they're not doing as good a job as this person did. But the point is that they made a decision what to leave out. And so they were making editorial decisions to edit, and yes, to edit. Yeah, this is pushing it a bit, but I'm trying to make the point that you can overdo, you just put too much stuff in your figures. You have to reconsider this. So I'm going to sort of end with what I think about, because it would be fun for some of us to have a conversation perhaps about this. Ultimately, everything I do in the science world is maintaining in the photographing science is I absolutely have to maintain the scientific integrity. I cannot do stuff to images, and you can't either. There are times when you can, if you indicate that you've done so. So in this situation, I made this picture of these are fluorescing gels that have these are gels that have absorbed fluorescing material. And I made the image on film. Maybe some of you have heard what that is. And it turns out the film did not capture the orange wavelength. What my eye was seeing was this picture. And so the image that I made was a misrepresentation. This digital, I scanned it and then digitally colored it. This is what my eyes saw. This is what the researcher wanted us to see. When I discussed this with the editors of Nature, they said to me that they would not permit this image for publication. I said, this is a while ago. I'd like to think that this might have changed. They felt that the this this original image had to be shown and not this image. And this was basically, it's an artifact of the process. And I think they're wrong, but I I would say to you, it is something for us to discuss at some point. Here's an image I made of a yeast colony from Jerry Fink's lab. I was blown away of the beauty of this morphology. I mean, these cells are telling each other where to grow. That's pretty amazing. And that is, in fact, the morphology of this yeast colony is the data. And so I deleted the background because I wanted you only to pay attention to what this research is about. Jerry felt that I was deleting information. Initially, he did not agree with me. He said if it's in the Petri dish, then you sort of know its size. I said, well, you could put that in the caption. In any event, it did get the cover of Science, so he went along with it. But anytime I do anything, I indicate that I've done it. For example, here, looking at Proteus colonies, which I made an image of Jim Shapiro's work an eternity ago, but you see these cracks of the agar. I deleted the cracks because we were submitting it to for a cover, but I indicate that I've deleted them. Over the years, a couple of people have said to me, wait a minute, you deleted what might be interesting. There might be some science going on within those cracks. And absolutely, that's right. But for this particular submission, it was about the pattern. And so I considered these cracks outliers. And so I deleted them. And so taking this to another step, as we all agree, deleting outliers affects what we see. If we delete the outliers, the question is, are we deleting important information? Are some outliers more important than others? Can we teach the machine to decide? And here I'm getting to the whole issue of all of AI now is all about teaching the machine. And are we teaching the machine properly? Are we biased in our teaching? Quite a while ago, David DeRosier created this amazing image with 100 SEMs. 100 were combined. And in of flagella. In the article, it says, I'm repeating this, the motor proteins do not survive the extraction procedure and so are not shown. He considered that those proteins were outliers. What happened was It is so incredible that the flagella looks like this, almost so beautifully perfect, that a number of organizations used it as proof that there is an intelligent designer, that it had to be done by an intelligent design. We have to be careful about how it is that we make images and how we tell people about how we make the images. So, for example, when we are looking now at the Webb, the James Webb images, and we are blown away of the beauty of all. This is extraordinary. And Hubble was also extraordinary, which I'll show you in a second. But these Hubble, these Webb, James Webb, oh my God, it's doing things that we never saw before. But people actually think the universe looks like this. It doesn't. This specular star is an artifact of where the tiles are attached, or come together. And that's why we're seeing this artifact, and we think that the universe is showing these. No, it's not. We have to tell it. We have to tell the public what we're doing and why you're seeing what you're seeing. In addition, the tools we use also affects what we see. So during the previous Hubble images of this particular area, let me just it's the pillars of creation. That's right. I had to look it up. That was from Hubble, and this is from the James Webb. It's even more amazing. We're seeing more detail. Look at all the stars back there, and we didn't catch it with among other things. It goes on and on. Now these were colored properly for reasons. I mean, they talk about it, but they don't tell it right up front. They're telling it on the web more than they used to. This is using near infrared. This is visible optical, but it's pretty fantastic. But I think we should know. I think we have to have the responsibility of showing how we get these amazing images. John Berger wrote in Ways of Seeing, we only see what we look at. To look is an act of choice. My hope is that some of you will be interested in doing what I'm doing, and that you help us spread the word of how amazing science and engineering is in research. And I'm happy to hear from any of you, and I mean it. Thank you very much. We're done.
If you have any questions, please, you can speak out or you can send a chat, whichever way you like. I have one question, Phyllis.
Yes, of course.
On the covers, I think not you, but on the covers of magazines, we see fantastic pictures, but they actually also give a description of the picture, uh, what is on the front cover. So, do you think that description uh, of what is there on the front cover is an addition to the meaning that the picture wants to depict, or is it only just for say academic interest?
Are are you asking, should we include more information about how the picture was made?
Yeah.
Yeah, I think that's a great question, but they're not going to do it. Because there's no, it's a wonderful idea. I haven't thought about that, but forget it. Unless all
I
of us,
thought actually, if you want to look at a picture and imagine what's going on there in a field of science or whatever it is, then you need to know the science very, very well, that particular area where the publication is focusing on, you must know that area very well to understand that straight away without any any explanation or anything like that.
Yeah. No, I feel strongly that we should give as much information as we can. I think people are interested in process. I think I know that I am. Um, but wouldn't that be something? If actually, let me quickly tell you if any of you are interested. The Economist is a British, very fine publication. They have how we got the cover. They tell you the ideas behind their covers. Their covers are mostly metaphors. I'm using a number of them in this third book called Abstraction, which I didn't talk about. But they talk about we started with this idea and then we decided to go to this idea and they talked about why they decided on the final cover. And I, you know, they're showing it. And so maybe people are interested. Maybe I'm wrong, you know. Let's see, there's a question. Thank you for, oh, thank you. You liked what you saw. What programs? Yeah. I basically I use Photoshop. And I don't use all of it. Photoshop is very, very complex. I'm actually not permitted to use more complex software in a way, unless it's an illustration. You know, when I put those covers together with pieces, I did it in Photoshop. Um, so that's what I use.
I see. And can you actually,
Felice,
Yeah.
um,
yeah,
I have a question. This is Mary Virginia Orna
Hi.
from the East Coast. Hi. Um,
Oh,
Felice, I once photographed and then had one of my images placed on the cover of an issue of the Journal of Chemical Education.
uh-huh.
And then when everything went digitized, I found out that none of the covers were preserved. So that in the e copies of the journals, there's no cover. And I'm wondering if that is the fate of the e copies for these journals as well.
Um, well, the answer is no, it's not. It's not the fate because they're all if you do a search for old science or nature covers, they've done a terrific job. But yours was fairly recent. Mary, when are you talking about?
No, I'm talking about maybe um uh maybe 20 years ago.
Yeah, but
Um
20 years ago, you're telling me you're telling me they actually did not keep your picture?
no, I'm telling you that when you go to find copies
Oh,
of of the Journal of Chemical Education online,
yeah,
that there are no images, there are no covers,
yeah.
that you cannot find a cover um, for each issue.
Yeah, that is very unfortunate, and that has to do with them because most of the other journals, as I mentioned, if you go online, you could see covers from 25, 30 years ago. And you should, well, do you have hard copy of it? Do you
I
have the,
used to, I used to, I had a collection. Um, but
oh, I'm so sorry to hear this. It's that
yes,
they did you write to them?
no, I just assumed that this is what they do when they make an e-copy of the journals and so forth, that they don't preserve the covers.
I don't, I have to tell you, I think it's worth asking them. They're good people. And because, again, when you go to other journals, you will see covers. In fact,
All
you
right.
could,
Well, I'm glad. I'm
yeah,
glad for
absolutely.
you.
Thank
But
you. Yeah, no, yeah, they should absolutely include your work in the digital files. They absolutely have to do that.
yeah,
I'm so
well, they're not there. They're simply not there.
That's awful, awful.
Yeah.
I'm sorry about that, really.
Well, I'm not sorry about it, but I was fearful for what you're doing
Oh,
and
thank
hope
you.
that you don't experience that as well.
Oh, thank you. No, so far, so far they all exist. Thank you.
Good, because they're magnificent
Thank you
and
so
really, really very instructive.
Oh, good. That's the nicest thing you could say, Mary. Thank you for that. Thank you.
Thank you.
Okay, it looks like there are no more questions.
If there are no more questions, then it's my uh, responsibility, let me call it that way, to thank Phyllis Frankel for a wonderful seminar. Very, very interesting seminar linking photography to different types of STEM subjects, actually. And one of the phrases that she has used in one of those magazines, capturing science to capture attention, is, is the motto of all this work that a photographer does, I think. So we are definitely very impressed with her work in trying to uh, convey the message across of a scientific topic by capturing science for our attention. Thank you very much, Phyllis.
Thank
Thank
you,
you.
everybody. Thanks
Yes,
so much.
thank you. Thank you. Applause. Thank you.
Thanks, Mary. A pleasure. Thank you.